Question number 1. In the beads on string model, the beads is made up of 6 histone proteins, 8 histone proteins, 6 histone proteins and DNA, 8 histone proteins and DNA. Correct answer is option B, 8 histone protein. The beads on a string model is for the nucleosome. It consists of the 8 histone protein core and the bead and the DNA found around the emitting string. Question number 2. How many types of histone molecules are found in nature? 4, 2, 5, 3. Correct answer is option C, 5. Name is H1, H2A, H3B, H3 and H4. Question number 3. Nucleosome is made up of DNA histone coat protein, DNA histone coat protein linker H1, RNA histone coat protein, RNA histone coat protein linker H1. Correct answer is option B, DNA histone core protein and linker H1. The core histone proteins are H2A, H2B, H3 and H4 over with the DNA is wrapped. Histone H1 is not a part of nucleosome core particles. Instead, it binds to linker DNA and thus referred to as a linker histone. Thus, the histone core, linker histone and DNA are components of nucleosome. Question number 4. With respect to assembly of every core histone, which of the following is wrong? A conserved region, histone fold domain, disc-shaped structure, 2 alpha helix and unstructural loop. Correct answer is option D, 2 alpha helix and unstructural loop. The histone fold is composed of 3 alpha helix and 2 unstructural loops. In each of these cases, histone fold mediates the formation of head to tail heterodimers of specific pair of the histone. Question 5. Which of the following histone pair form tetramers in solution? H1 H2A, H2A H2B, H2B H3, H3 H4. Correct answer is option D, H3 H4. H3 and H4 histone first forms heterodimers, then they come together to form tetramer with 2 molecules of each. In contrast, H2A, H2B form heterodimers only in the solution and histone H1 only act as a linker histone. 6. Association of DNA and histone is mediated by covalent bonding, hydrogen bonding, hydrophobic bonding, van der Waals interaction. Correct answer is option B, hydrogen bonding. 7. Which of the following facilitate nucleosome positioning? Nucleosome remodeling complex, topoisomerase 2, SMC protein, DNA binding protein. Correct answer is option D, DNA binding protein. 8. Which of the following region promotes histone DNA association? AT, AG, CG, CT. Correct answer is option A, AT region. 9. With respect to N-terminal modification of the histone tail, which of the following option is true? Lysine phosphate modification, serine methylation, acetylation transcriptionally active modification, involvement of gene expression. Correct answer is option D. Acetylation marks transcriptionally active region, where methylation makes both transcriptionally active and repressed region, and phosphorylation histone H3 commonly seen in highly condensed chromatin. Thus, this modification result in a code that can be read by the protein involved in gene regulation and expression. 10. Which of the following is a function of modification histone tail? Association with adjacent nucleosome, interaction with DNA backbone, interaction with chromodomains, formation of 40-fold DNA structure. Correct answer is option C, interaction with chromodomains. 11. Which of the following enzyme of transcription commonly contains a bromodomain? TF2D, CDK, pre-RC, DNA polymerase. Correct answer is option A, TF2D. Explanation. Bromodomain containing proteins are involved in regulating transcription and formation of heterochromatin. Only transcription factor 2D is of the above option is involved in transcription of DNA, whereas others are involved in DNA replication. Question number 12. Which of the following enzyme is not correctly paired with the function? Acetyl transferase add acetyl group to lysine. Correct. Methyl transferase adds methyl group to serine. Not correct. Is the answer. Topoisomerase association with nuclear scaffold. Correct. Deacetylase remove acetyl group from the serine is the correct. Answer is option B is the correct answer, which is not correctly paired with their function. 30. Which of the following is not promoted by histone tail modification? Formation of repressive structure, gene expression, nucleosome remodeling, nucleosome sliding. Correct answer is option D, nucleosome sliding. Nucleosome sliding is a type of nucleosome remodeling. It is facilitated by nucleosome remodeling complex. It cannot be promoted alone by modifying histone tail unless a nucleosome remodeling complex come along. 40. The extent of chromosome coiling in non-dividing cell is supercoiled euchromatin, condensed heterochromatin. Correct answer is option B, euchromatin. 50. Why a chromosome condense? To facilitate accommodation, always condense to facilitate cell division, to facilitate distribution in daughter cell. Correct answer is option D, to facilitate distribution in daughter cell. 60. At which phase do transcription cease? Interphase, prophase, S phase, G phase. Correct answer is option B, prophase. Cease यानी कि stop. 70. The part they play a critical role in even distribution of parental DNA during division is telomere, centromere, spindle fiber, centrioles. Correct answer is option B, centromere. 80. With respect to centromere, which of the following is wrong? Constrict chromosomal region, holds the sister chromatids together, attached to spindle fiber, facilitate even distribution. Correct answer is option D, facilitate even distribution. 90. Centromeric DNA was initially defined in bacteria, fungi, yeast, human. Correct answer is option C, yeast. 20. Telomere is not related to maintenance, chromosome degradation, division, replication. Correct answer is option C, division.